Now I will invite Dr. Ifra Ghori, who's a lecturer at Fatma Jinnah University, and she's a dear comrade of mine. Welcome, Dr. Ifra.
Thank you so much for giving me opportunity to present on this webinar. It's a very wonderful opportunity. Here I would present my research work which I conducted in my PhD, and that is basically related to the use of indigenous antibiotics for enhancing the productivity of Labeo Rohita through gut modulation for sustainable aquaculture, a metagenomic approach. So starting with okay. As all of you know that fish is very important in human diet because of the protein it carries. It's very digestible and it has got the essential amino acids, and it's very good for the heart health, and it is rich in omega-3 fatty acids. So this was the rationale to use this this model organism in my research. Uh, you know that the fish capturing techniques have been shifted towards farming. The farming is raised because of the depletion of these wild resources and poor capturing practices, as well as the environmental pollution, such as these contaminants, mercury, PCBs, pollutants. They are major concerns for the wildlife. This has shifted the trend towards the farming. And one of the another very important reason is the growing human population. That is why the farming is on demand. Now, aquaculture, what is aquaculture? Aquaculture is breeding, rearing, harvesting of animals, plants, both in fresh and marine water under the controlled conditions. Probiotics, as you all know, they are living organisms. They have extra nutrition, beneficial effect to its host. That concept was first launched by Ali Mashnikov, and later on it was improvised and it was worked on by a lot of successors. Initially, the probiotics were used by Japanese yield in order to enhance the growth and not to reduce the mortality. Then a lot of commercial preparations were made for enhancing the bacteria, for enhancing the growth in the bacteria. They were mainly from the bacillus and other species. And probiotics basically are environmental friendly and cost effective approach. Now, there are a lot of benefits of using the probiotics. They can produce a lot of antimicrobials. They produce digestive enzymes. They modulate the microbiology. They enhance the growth, and they have a very good impact on the physiology. I've worked on the Labeo Rohita because it's very common in South Asia. It's economically very feasible, and I selected it because of the high consumer demand and quality and of its fast growth. Then, first of all, I isolated bacterial strains from fish gut, and then I did biochemical characterization. I also selected some of the strains which were previously identified from dairy source as well as from silage source. And then I characterized them for the probiotic screening. These tests included bile tolerance, acid tolerance, antimicrobial potential, antibiotic susceptibility. And after doing all these tests, I selected 9 treatments, uh, including the commercial treatment as well as from the different consortia, as well as single mode based probiotic was included. Then they were actually, I did a 90-day feeding trial to the fishes in which 9 feeding groups were made. And then after that, I checked different parameters, which include hematological parameters, the growth parameters, as well as I also did the microbiology of them, both on culture dependent as well as the culture independent methods. The study which I conducted after 90 days, I found that the core phyla, which is found in all that treatment, there were 9, basically 9 probiotic based treatment. 1 was control and 8 was probiotic based treatments. The core phyla, which was found in all the treatments of probiotic, were Proteobacteria, Firmicutes, Bacteroidetes, and Actinomycetes. Dominant bacteria was found to be Galactomycetes. And I found that the mixture of G. candidum and Bacillus was found to be very, very good for the fishes in terms of the growth and as well as for their FCR and hematological parameters. One of the things which I noted during my research was that the potential pathogens of the fish was absent in nearly all the treatments, especially those treatments in which the fishes were fed with the probiotics. Like the pathogens which is Sporobolus, Myces, Leptococcus, and Trichosporin and Cytococcus, they were found in a very low percentage in all the treated treatments. So probiotics here, after the results, it was shown that they modulated the microbial community by changing the level of microbial population. Geotrichum candidum was established in the fish gut, and it was indicated by the increase in their percentage in T1 treatment and T7 treatment, as 9 treatments were given to the fishes. And these treatments will also seems to be effective in reducing the potential pathogens such as Staphylococcus aureus and beneficial bacteria number rises as Achromobacter also rises when I saw the metagenomic analysis of that experiment. Achromobacter, which emulates the nitrogenous waste and maintain water quality for the better fish cultivation. Opportunistic yeast such as Trichosporin and Cryococcus was also found in a very low percentage in control as well as in the treated groups. So probiotics, when they were applied both in single and consortium, they showed significant impact on fish physiology when they were fed at a certain concentration. Probiotic supplementary treatment shows significant influence on growth rate, weight gain, feed conversion ratio, feed efficiency, which results in better fish growth. Significant impact was shown on hematological parameters such as RBCs, WBCs, HGB, HCT, which depicts that the health status of the fish. GIT enzymatic activity was higher in treated groups as compared to the control, which is better digestion. Probiotics in consortium form showed synergistic and more significant results. The potential probiotic feeding modulated the gut microbial composition of Labeo Rohita fish. Treated groups showed reduced number of OTU species, and that could be due to the competitive exclusion and antimicrobial potential. The group which was treated with geotrichum, both in single as well as consortium form, showed higher reads of geotrichum, which reveals its survival in the fish gut. So geotrichum 01 dominated in treated and the control treatment. The core phylum, which was found after the metagenomics, it was found that the core phylum that was found in all the treatments, including Proteobacteria, Firmicutes, Bacteroidetes, and Actinobacteria. Probiotics that were used in trial modulated microbial community by changing the level of the microbial population. And the geotrichum was established there. Opportunistic pathogens were not present when I analyzed the metagenomic data. The overall potential pathogens percentage was lower, and treated group shows the lower level of Staphylococcus and Clostridium and Listeria. And when I exposed the treatment, the groups, the best performing treatment to the challenge test, then no mortality was observed during the whole trial, as well as during the trial which was subjected to the Staphylococcus aureus exposure. Aromonas hydrophila in the potassium pathogen was not detected in any treatment. So the overall pathogens, they were not found during the metagenomic data. So it was found that by using the indigenous probiotics and by use of this next generation genomes sequencing, we can modulate the gut microbiota for the better health and growth of fish and for the better productivity of fish. Thank you so much.
Thank you, Dr. Ifra. It was such an interesting talk based on your PhD research work, and it was an utmost pleasure hearing you. Thank you very
Thank
much.
you so much. Thank you so much.
You're welcome.